Actually, the antibody maybe in the end of the 19, I think it's 19, the end of the 19, the antibody did not found, but so many experience to discover it and research it. So at first we will introduce some history of the research about the antibody, including something about the funding and further research. Here we can see the PPT is described the process of the discovery of antibodies. The Louis Pasteur is a very famous person in our immunity. He in his experiments on the sheep and with a conclusive trial on human, demonstrated that vaccination was able to confer immunity. The clue for the role played in antibody in conferring immunity came from the experiments of the Emil von Behring and Sabiro Cassetto in 1890. Yeah, 1890. In the 1890, they concluded serum from guinea pigs that were previously exposed to Caroline bacteria. Diphtheria, the bacteria that caused diphtheria. This serum was injected into a live animal and found that the receptors remained resistant to diphtheria. From the experimental results, they inferred that a factor was responsible for protection. So subsequently, various researchers designed experiment to search for this protective factor or agent that conferred immunity and demonstrated that an active component of the serum could participate toxin, neutralize toxins, lyse bacteria, or aggregated the bacteria. And in each case, the protective factor or protective agent was differently named as participating ectotoxin, antitoxin, and bacterial lysis and agglutins, respectively, based on the type of the activity. In this context, since the immunity was mediated by the antibody in the body fluids, it was called humoral immunity. So here is some point about the history of discovery the antibody. Here is a diagram about the Emil von Behring and the antitoxins. Here showed this man is the M. von Behring, and he in this experiment he used the tetanus as a toxin to inject it into a horse It means immunize horse, and then get the serum from the horse. Furtherly, inject the immune horse serum to a patient at a risk of tetanus, and the final, the patient was protected. So he gets that there was something existent in the immune host serum to against the tetanus, and as a result, the immune host serums protected the patient. What is the substance existent in the serums? Today we know it, call it antibody. But in the process to discover it or research it during so much difficult and imaginate process. So every achievement in this process, it has put the step forward and push, make so many progress. So every excellent work, they got the Nobel Prize, such as the Bering and the Herlich and the other person. So this class we majority this class we mainly to introduce the 6 points about the antibody. The first one is the definition of antibody and the basic structure of antibody. And the third one is the biological characteristic of different antibodies and biological activities of the antibody and the generation of antibody diversity. And the last, the monoclonal antibodies. Okay, one by one to introduce it. So the definition of the antibody here is a sentence to describe it. Antibody is a globulin which is produced by plasma cells as a result of the induce of antigen, and which has the ability to combine with the antigen that stimulated its production. Actually, this sentence is described a two-step of the process. The step one is the antibody or the foreign substance active the B cell. The B cell recognizes the antibody or recognizes the foreign substance, maybe the special epitope, right? B cell recognizes the epitope and then differentiated to the plasma cell and produce the antibody. This is the first step. And the second step is the function of the antibody to perform. The antibody will combine with the epitope on the surface of the antigen. Like this, the antibody will to display the function. So the aim of the antibody production is to combine with the epitope. So we can see another norms here. The B cell. The B cell recognizes the antibody or recognizes the foreign substance, maybe the special epitope. But B cell recognizes the epitope and then differentiated to the plasma cell and produce the antibody. This is the first step. And the second step is the function of the antibody to perform. The antibody will combine with the epitope on the surface of the antigen. Like this, the antibody will to display the function. So the aim of the antibody production is to combine with the epitope. So we can see another norms here. Globin. Globulin. Globulin. What means the globulin? It's a globulin. Some globulin named antibody. It means another part of the globulin is not antibody, right? So here is shows the some information about the globulins. In 1930s, the Cabot found that the serine in the serine, so many globulin in the serines, and in his mind, the gamma, the group of globulin named is globulin named gamma globulin. He called the immunoglobulin. Immunoglobulin. The gamma globulin is he named the immunoglobulin. Actually, in the fact, his experiment is not totally right, because some alpha globulin or beta globulin also contain some immunoglobulin. So this result is maybe 90 years ago, right? 90 years ago is not I think it's some wrong or some I think it's some wrong the communication in this part. So what I want to say is we should distinguish the relationship with antibody and the immunoglobulins. Maybe in sometimes we call the antibody as immunoglobulins, but actually the antibody is immunoglobulins, is right, but we couldn't say the immunoglobulins are the antibodies. It doesn't work on the other way around, because the antibodies is the antibody, because the immunoglobulins contains another globulins expected antibodies, like the picture showed here. The Ig type receptor family. In the structure level, the receptor showed in the picture in the red box here shows the Ig type receptor family. This protein in the structure is like a globin, and his function is relative to the immune response, but it's not an antibody. Just structurally, just similar on the structure level, but the function is not to immune response. So we talk about the antibody here is a special part of the immunoglobulin. Maybe if you read some paper or literature in the future, you will see the antibody and the immunoglobulin identical to use, but you must understand the difference between them. Like the diagram showed here, the antibody is the subselection of immunoglobulins. Yeah. And the next, we talk about the basic structure of the antibodies. This part we should mention the two persons here. The one is Eldman, and the other one is Porter. The Eldman he was supposed that the molecular of the antibody is very simple and contain the disulfide bondings. So after his experiment, he obtained the heavy and light chains from the antibody, and he also found that the single heavy chains or light chains didn't work in the immune response. And on the other hand, the Porter have another result from his experiments. He used the pattern to treatment with the antibody, and got the FAB and FC fragments. So together, the two persons results, they posted the structure of the antibody molecular. Is the shape of Y, right? Yeah. Due to the excellent work about the antibody structure, both of them got the Nobel Prize in 1972. So here is another kind of diagram about the antibodies. It's more complex than the before. The general structure described below pertains to IgG, the silent silence feature of different class have been subsequently described. We can see in the diagram 4 polypeptide chains, 2 light chains, and 2 heavy chains. And hold together by the disulfide bonds. In this molecular 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 15 disulfide bonds in the molecular of antibody, and this molecular will show the Y-shape structure. The amidogen terminal it including as 2 terminal. The one is the amidogen and the carbasco terminal. Sorry, today someone around in around or behind my seat. Some trouble about some trouble to my expression. I'm not feel good now. Okay, go. This is the basic structure about the antibody. And the heavy chains composed of about 500 amino acid, and the light chains including about 200 amino acid. The IgG is a globular glycoprotein that has a molecular weight of 150 kD. It makes up of 4 polypeptide of two different kinds. It is composed of a pair of identical low molecular weight chains called light chains, and a pair of identical high molecular weight chains called heavy chains. Here shown is a structure as a special structure of the IgG after formed to a special structure. The L chain has the molecular weight of 20 and 25 kD and consists of 214. Well, the heavy chains have a molecular weight of 55 kD, and they make up about 440 amino acid for each. The light chain of most vertebrates are shown to exist in 2 distinct forms named kappa and lambda. We can see the many beta sheet, and to form the substructure. Those substructure is very important to keep the function in the immune response. Actually, is main response to epitope. So here showed is the detail region about the antibody. In the red box, here is identical parts V region. It means the variable region. The left and the right, the sequence and the structure are identical. And the below of the V region, and the rest of the antibody structure is molecular is C region, the consistent region. And actually, the V region also also have another detail structure. It means CDR is a complementarity determined region. We already knew the V region is responsible for the recognition of epitope, and the CDR is the I think is the point to touch with the molecule on the antigen. Antigen, yes. How the people or how do we know the CDR exist? Because this test so many different kinds of antibody or the light chains are sequenced and put them together to compare so many different kind of light chains put together to compare each other. They find out 3 key regions have a high variable frequency here. Here marked as the CDR1, CDR2, and CDR3. And another part of the light chain is a small variable variation, smaller level of variation between each other. So you should be understand here. This picture showed as so many different kinds of light chains together to compare with each other, and got the data, showed the key point for some different each other. So the CDR 1, 2, 3 maybe the variable key point to responsible recognize the epitope. So here we show the three kind of CDR. It's like a finger, maybe three finger on the left chain and three fingers. I think it's fingers on the left hands, or the three finger on the left on the other hands, and six fingers together to get the epitope, just like the finger to get an orange, right? Different and together at to get the orange is very well to get it. So I think this picture is very clearly to describe the specificity for the antibodies V region to recognize the epitope. AG antibody. Here the AG means antibody. Antibody recognizes the one epitope of antigen and bind to V region. And L1, 2, 3, and H1, 2, 3 means the three CDR in the L means light chain, and H means heavy chain. Three CDR in the different chains, two together, and high are the hypervariable region called CDR, that the key site for the bonding. Hypervariable region provide possible for unlimited variety of antibody to bind diverse epitopes. Besides the V region and the C region, another special region hidden in the C region. Here is named the hinge region. This region is very small and very maybe less than 10 amino acid. But this structure is very important for the function of antibody, because this structure, the huge region, is provide a special property such as flexible, because it reach proline. The function of the hinge region is to facilitating the interaction between antibody and antigen, and facilitating complement fixation. We can see here the IgG, for example, the IgG molecular conformational change induced by antigen binding. We can see the left diagram of this PPT. The antibody did not combine the antigen, so the conformational structure like this, and the C1q protein is a protein to active the complement system. The complement system we will talk about in the future lessons. Before the binding, the CQ1 binding site is closed. It's closed, just showed in the left diagram. But on the right, after the binding of antibody and antigen. The conformation structure of the antibody changed obviously, and the C1q binding sites were exposed. So this change will promote the CQ1 to binding here, and next, the complement system will activate. So in the immune response, the process of the conformation change in the IgG after the antibody to bind with the antigen, the very key point for the immune response, because this change is active the complexity system. And I think we should know some norms from another levels or accept, especially the norm of conception of the domains. The antibody is a very small molecular, but we also describe the structure, use the demos. The demos domains, the domains is the polypeptide chains followed by diffuse field bound into a global region. Here, I use the red arrows marked here. VH and VL is the domain for antibody binding site with the epitope. We can see the picture. Each light chain has two domains. One variable domain and another consistent domain, CL. Well, the each chains has four domains. One variable domain, VH, and three consistent domains. The heavy chains of IgA, IgD, and IgG have three CH domains, and the huge region between CH1 and CH2. In contrast, IgE and IgM have four CH and do not have a huge region. So the IgE and especially pay attention here, the IgE is a very special antibody. We will talk about it in later. So the carboxyl terminal domain in IgA, IgD, and IgE is designated CH3 and CH3, where the IgE and the IgM is CH4 and CH4. The third part, the biological character of different antibodies. Different class of antibody vary with the respect to the count of the heavy chains that are associated with the L chains. In IgG, for example, the heavy chain of IgG are of gamma type. Depending on the light chain are the present. The IgA, the heavy chains are alpha chains. In IgD, there are delta chains, and in IgE, there are optional chains, and the IgM is the mu chains. In any one molecule, the two heavy chains are identical to each other, and the two light chains are also identical. Further, IgG, IgD, IgE occur as a molecular monomer, and the IgA is a dimer, and the IgM is a pentamer, and the pentamer. We will introduce the four class of antibody one by one. The first one is IgG. IgG is a monomer and with the very special properties. IgG is one of the major antibody in the serum. It accounts for 75%. And the IgG is the longest half-life. The half-life is 23 days. And this antibody is the major IgG in the extravascular space. And the IgG is the only one antibody to transfer from mother to the fetus. Only one. Only this kind of antibody can transfer from mother to fetus. And the function of the IgG to perform is binding to the cell, which named IDCC. We will also talk about the IDCC is a very key process for the antibody to perform its function. We will be talk about the IDCC in tomorrow on tomorrow. We'll talk about the IDCC. Here is the IgG. And the second one is IgM. IgM has two kinds of structure. The pentamer, five monomer together linked with a J chain, joining chain together, and the membrane band type IgG, which here name is MIgG. Here we showed the monomer IgG. It's like a tree have a root, right? Have a root insert to the membrane and locus on the surface of the cells. The joining chain, joining chain is a very small peptide and used to link the monomer. As a result, 5 monomer IgM together to form the pentamer, and the 2 IgA together linked by the joining chains to form the dimer. Actually, the IgA, the SIgA accepted the joint chain. It also have a secret piece. The secret piece only existed on the SIgA, which has a special function, such as protect the SIgA from desiccation by other enzymes. And helped the SIgA transferred across the cell membrane. And the property of the IgM, it is the first first antibody made by the fetus and B cell, and phagocytation complement is active classical pathway for the complement system. And due to it the pentamer have 5 IgM together. So the molecular weight of the IgM is the largest. So it is the largest size of molecular. And it also performed ADCC. ADCC, it is the same to the IgG. And the IgA is described before, and the just the how do you say, whatever. The IgA is including the dimer and the monomer. The dimer will exist in the serine, and the dimer will secreted to the outside of cell. The IgA coccus in the mucous secretions such as saliva, tears, and nasal fluid, sweat, milk, and another fluid. And the IgA is synthesized locally by plasma cell, and before it is secreted, it is dimerized intracellularly together with cysteine. So here we also introduce the special structure of the secretory piece. And here is just I said before the function of the secretory piece. And the function of the IgA is to transfer to the neural through colostrum, and the important antibody against the mucosal infection. So the IgA is the only one antibody will transfer out of the cell. and is the major antibody to secretion. Also performed the function on the mucosal systems or mucosal tissues. And the IgE is also a very special antibody in our in the body, because the concentration of IgE is very low. Here described the least common Ig antibody, the least common antibody, and the property is a very special property of IgE is binding to basophils and mast cells, because this binding is lead to the hypersensitivity, the one type hypersensitivity. Here is a diagram about the hypersensitivity. First, the one type, because the IgA is have a high affinity to the receptor on the master cells, and this binding is I think it will provide a chance for the master cell to release the special chemical substance. The release of glycotrates and histamine promotes strong stimulation of vascular tissue fluid exudation and promote inflammation. So the produce of IgE is not a good thing for our body. So many persons suffering from the one type of hypersensitivity due to some foreign substance or foreign antigen induce the body to product the IgE, and the IgE bind to the master cell, and the master cell release the glycotrains and histamine, and as a result, the person is suffering so many painful. I think is not a good thing. And the last, the IgD. IgD is one of the surface of mature B cell serving as BCR, mutual marker of the B cell. So we distinguish the B cell mature or not mature. We should check the molecular of IgD. And actually, there are almost no. It's hardly to detect the IgD in our blood or our serum. Almost of them are located on the surface of the B cell. So the IgD is I think is not good, is a not important antibody in our body in the immune response, but maybe the function of the IgD is not totally relieved. And the last, let me see the distribution of the antibody. Here is a description. And we can see the diagram shows that, and the IgM is in the blood, and IgG in the tissues and blood, and the IgA almost in the mucosa, and the IgE in the surface of our body, such as the sweat and IgA. And the sliver, the IgA, is existed in the sliver, teams, and sweet milk, and another fluid. So the major antibody is the IgG, and the IgA is a very special antibody because it is the only one antibody can secrete from our body. Okay, this class, the content of this class is over. I think there's something wrong with my data. So anyway, have any questions? Okay, class over.
好的，老师，谢谢。
Thank you,
谢谢老师，
Thank you, 老师. See you again.
Welcome. Okay.
再见。
再见。